చేసా హలో ఏంటంటే హలో Sir, good morning, sir. Mohan Varma, sir.
Hello, sir. Hello, how are you, sir?
Fine, sir. Fine, sir. How are you, sir?
Yeah, I'm fine.
ఒకసారి షేర్ చేయండి సార్ ఒకసారి.
Slide sharing, I'll do it, sir, once, sir. Okay, right.
Okay, sir.
I'll do
Okay,
it once.
sir.
Slide sharing, I'll see.
Okay, sir. Okay, sir.
So, in the outlet sharing, you can see some options available there. You can see once. And the host ninja outlet annaru. Here is giving that actually host has not enabled the sharing another cherry. What's the option?
హలో సార్, మీవి యాక్చువల్ గా రెండు వస్తున్నాయి సార్, రెండు మొబైల్స్ చూపిస్తున్నాయి ఇక్కడ.
అదే సార్, ఎందుకంటే ఒక దాంట్లో నాకు యాక్చువల్ ఆడియో ఉండదు సార్, ఒక దాంట్లో నాకు ఐఎమ్ నాట్ బిహాంగ్ ఆడియో ఇన్ వన్ అనమాట అనుకుని. So I have to operate two anamata. Actually, in the case of one, I'll be seeing the audio. Okay, in another one, I'll be seeing then what slides and things.
Sir, now you check, sir, once. రెండిట్లకి ఆప్షన్ ఇచ్చాను నేను. సార్ షేర్ అవుతుంది సార్, అది జూమ్ అని వచ్చింది. పీపీటీ షేర్ చేయండి సార్.
వస్తుంది సార్. ఓకే
ఓకే.
Okay, sir.
Okay,
oh, sir, are you able to see the slide, sir?
Visible, sir. It is visible. Audio also clear, sir.
Audio is clear, no, sir? Okay, yes, sir. Good, sir. Right. Thank you, sir.
Just wait another 2 minutes, sir. 2 minutes.
Okay.
So by 11 we will start, sir.
Okay, sure, sir.
హలో, నో ప్రాబ్లం. ఓకే ఓకే. అదేనా మీరే ఇప్పుడు కాల్ చేస్తున్నారు, కట్ చేస్తున్నానండి. నేను జాయిన్ అవుతున్నానండి.
Hello, sir. We can start, sir.
So, good morning, delegates. Once again, this is the fifth day of the FDP, exploring new innovations in all drug delivery systems. So, today we have a guest from Sri Vishnu College, Dr. M. Mohan Varma Garu. So our principal sir, Dr. V. Bhaskar, is presiding the meeting. Yes, please. Sir, please proceed, sir.
Good morning to all the participants and delegates. I am Dr. V. Bhaskar Raju, Principal and Professor of Sri Vasavi Institute of Pharmaceutical Sciences, and I am delighted to organize 1 week online national faculty development program on exploring new innovations in novel drug delivery systems. So welcome all for the day 5 of this session. And today's research person is Dr. M. Mohan Verma, Professor and Dean, Sri Vishnu College of Pharmacy, Bhimavaram. On behalf of our management, organizing secretary Dr. Narayan Raj Garu and organizing committee, I welcome our resource person to share his valuable insights in novel drug delivery systems. Now I request our faculty, Pallavi, to give a brief introduction about our resource person, Dr. M. Mohan Verma Garu. Pallavi, you can proceed.
Good morning, delegates. Thank you for registering for FDP conducted by Sri Vasavi Institute of Pharmaceutical Sciences, Tadepalligudem. I am glad to introduce our today's distinguished speaker, Dr. M. Mohan Verma sir, who is currently working as a professor in Sri Vishnu College of Pharmacy, Bhimavaram. He completed his B.Pharm in Berhampur University in 1989 and stood as an university topper, and completed his master's and PhD in IIT Banaras Hindu University, Varanasi. He is having 26 years of teaching experience, and he guided 8 PhD scholars and 70 M.Pharm project students, and having 108 international and national publications. He was awarded Gandhian Young Technological Innovation at Rashtrapati Bhavan, New Delhi in 2018. He is a lifetime member of IPA, APTI, IPGA, and ISTE. He completed project on effect of cyclodextrins on the dissolution rate and bioavailability of rosiglitazone of 10 lakhs grant from AICTE. He completed olfacting deficiency in Indian population with cognitive impairment, a pilot study with 53.68 lakhs grant from DST. We are happy to have you here today, sir. Now I request sir to share his view on electrospinning technique. Sir, please proceed, sir.
Okay, sir. ఒకసారి చెక్ చేస్తాను, sir.
Thank you for the introduction. Thank you all of you, sir, for organizing this FDP and for inviting me for this FDP. I'm extremely thankful to you, sir. So I'll start the topic today, applications of electrospinning. So the brief introduction of the electrospinning: when a high voltage is applied to a liquid droplet, the body of the liquid becomes charged, and the electrostatic repulsion will counteract the value of the surface tension, and the drop is tested at a critical point. A stream of liquid erupts from the surface. This point of eruption is known as the Taylor cone. The elongation and thinning of the liquid stream leads to the formation of uniform electrospin nanostructures with nanoscale from the diameters. The nanofibers with diameters ranging from few nanometers to several micrometers can be produced using an electrostatically driven jet of the polymer solution or the melt. The word nano is obtained from the Greek word nanos, which equates to a dwarf or something extremely small. Technically, a nanometer is equal to 1 from the billionth of a, from even the structure of a, from even the meter structure. Thus, all nanoscale structures typically have a dimensional range or length between 1 and 100 from the nm. So you can see the electrospinning apparatus in the slide. Now here electrospinning, it can be given in the two ways. One is the horizontal type and the vertical type. In the electrospinning setup, mainly it contains 3 components. It contains from even the tube structure with a stainless steel needle. We have to apply the high voltage supply from 2 to 50 from when the kV volts has to be applied. And we can have a drum-like structure over which the electrospun sample will be obtained. So you can see in the slide the horizontal type of electrospinning. Here we can take the liquid sample. Either we can take an organic solvent or aqueous solvent along with the drug and excipient. And this can be sprayed over the drum-like structure. And here we have to apply high voltage with the help of from the syringe pump. In this slide, you can see the vertical type of electrospinning. Here also again, we can take the organic solvent or aqueous solvent along with the drug plus excipient. And we have to employ a stainless steel needle. The required high voltage has to be applied. And on the rotating drum-like structure, we can obtain the electrospun sample. So in this electrospinning topic, several from when the patents have been filed on the process of electrospinning. In 1969, Taylor obtained his work on the shape of the droplet produced at the tip of the stainless steel needle when an electric field was applied. Taylor found that the pendant droplet developed into a cone when the value of the surface tension was balanced by the electrostatic force and the jets were ejected from the vertices of the cone with the diameter smaller than the diameter of the needle when the electrostatic force exceeds value of the surface from the tension. In 1971, Baumgarten reported to make apparatus to electrospin the acrylic nanofibers with diameters in the range of 500 to 1100 nm. In 1981, and Mandley produced the polyethylene and polypropylene nanofibers by electrospinning their melt. The synthetic and natural from even the excipients have been used to prepare the electrospun nano from the fibers. So if you see the main applications of these electrospun nanofibers in the drug applications, electrospinning is simple. It is example is an efficient process. We can obtain the polymeric nanostructures, electrostatically driven jet of the polymer solution onto a drum-like substrate. So in the drug field, the electrospun nanofibers is having a very important application for the controlled release formulations. So the submicron nanofibers produced by the electrospinning has several advantages like tunable from the pores, high value of the surface area to the volume ratio. We can tailor the nanofibers with the desired composition in order to achieve the specific from the function. So the main advantage of this electrospun nanofibers are they are very small from when the size of pores. They have large area available. These from these structures having pores and which are the non-woven polymeric from the networks. They can be employed mainly so as to achieve the required from these properties. And here mainly the area can be employed. Here the area can be increased because we are obtaining thread-like structures of the nanostructures when you are applying electrospinning. We are spraying the sample onto a drum-like structure. We can obtain a very thin from even thread-like structures and the area available will be increased. So whenever you obtain this electrospun nanofibers, we have to adjust the thickness of these thread-like structures. So here we are having some important points like what is applied from the voltage, temperature, humidity, etc. By adjusting these points, we can exactly adjust the thickness of this from the thread-like nanostructures. So here the important points is absolute viscosity of the liquid phase, what of excipient you are employing, what is the value from this weight of the polymer you are employing, and how much applied voltage you are employing, what is from the distance from the stainless steel needle to the drum-like structure, what is the rate at which the feed is being injected, what is from even the diameter of the stainless steel, even the needle structure. Similarly, whenever you are doing the experiment, in the month of October or November, if you do the experiment, the temperature will be less and the humidity is less. In the month of April or May, if you are doing the experiment, the temperature and the humidity will be increased. So this can affect the thickness of from this electrospun sample, even the efficiency of the process. Suppose the equipment is completely, suppose if you install the equipment in some AC room, at the time we can exactly adjust the temperature and humidity so that throughout the year we can have the same efficiency of the process. So here, if you see the different from these factors which can affect the electrospun nano from fibers are, one is the molecular weight of the polymer. Here the diameter of the fiber increases with the molecular weight. Low molecular weight polymer will form the beads. So if you're employing the excipients having a very high molecular weight, then the diameter of the nanofibers will also increase. Second point is the absolute viscosity of the liquid phase. Whatever liquid phase you're employing, organic solvent or aqueous solvent, when you add the excipient plus drug, the viscosity will be altered. So here the nanofiber from thickness of the diameter increases as the solution viscosity increases. So both these points, if you increase the absolute viscosity of the sample, if you increase the molecular weight of the excipient, then automatically it will increase the thickness of the diameter of the electrosponge sample will be increased. Next point is that from this fiber diameter decreases, if we increase from this amount of the excipient drug within the liquid phase. Next, the influence of the surface from the tension. Electrosponge nanofibers without beads can be obtained if we are able to decrease the value of the surface tension of the solvent phase. So here in the liquid phase, if it is required, we can decrease the surface tension by adding the small amount of the SLS or the twin 80. So if we are able to decrease the surface from the tension, then without beads, we can obtain the very thin thread-like structure, so the electrosponge nano sample can be obtained. Next, the voltage plays a very important role. Voltage can be adjusted in the range of 0 to 50 from the kV. So the average diameter of the nanofibers will increase when we are applying the very high voltage. So whenever you are applying a very high voltage, automatically the thickness of the diameter of the electrosponge nano sample will be increased. So optimally, you have to adjust the voltage. Required optimum voltage has to be adjusted in the range of 2 to from the around up to some 20, it can be increased voltage. Beyond that, if it's required only, you can increase, otherwise not. Similarly, you have to also adjust the rate of flow of the solvent phase in which we have dissolved the drug plus excipient. Normally, if you take the diameter of the electrosponge nanofibers decreases as you are decreasing the flow rate. So whatever the flow rate we are having for the liquid sample from the syringe tip to the drum-like structure, suppose if we decrease the rate of flow of the liquid phase, then automatically the thickness of the diameter of the electrosponge nanofibers also decreases. So voltage we can increase at the time the average from this thickness of the nanofibers will also increase, and the flow rate if you decrease, automatically it will decrease the thickness or from when the diameter of the electrosponge nanofibers will be decreased. Next to the distance from the needle tip to the drum-like structure, when the distance increases, the average from the diameter of the electrosponge nanofibers also decreases. Similarly, humidity plays an important role. As the humidity increases, the thickness of the diameter of the electrosponge nanofibers will decrease. So whenever the atmospheric humidity is high, normally you can obtain a very thin structures. When the humidity decreases, the thickness of the electrosponge sample increases. Similarly, temperature also plays a very important role. Suppose if the value of the temperature increases, then it will actually decrease the thickness of the electrosponge nano sample. So here the various types of the natural excipients have been employed in the electrospinning process. Example is the chitosan, silk from this. So the various types of the excipients have been employed. Apart from this, we can also employ the PVA, that is polyvinyl alcohol, polyethylene vinyl alcohol, polyepsilon from even the caprolactone, polylactic acid, polyglycolic acid, and even the polylactic acid, glycolic acid from even this copolymer. So different type of the excipients can be employed in the electrospin process. Here, if you take the important excipient which is employed in the electrospinning widely from, if you take this from the chitin is obtained from the crab and the shrimp, and if you're employing the deacetylation reaction from if you take this from the chitin is transformed from when the chitosan, it can be widely employed in the electrospinning process along with the various types of the drugs. So here I'll explain what are the different type of excipients and what are the different type of solvents. So the aqueous solvent can be employed. Suppose if you're taking the celluloacetate as an excipient, here we can take the acetone plus the dimethyl estamide plus ethanol can be taken as solvent blend at a specific ratio. Suppose if you are taking from the chitosan as an excipient, we can take the pure water, ultra pure water along with acetic acid, because if you're taking the acidic pH in the aqueous phase, for example, the excipient from even this, if you take this excipient from even the chitosan, it dissolves easily. Next, ethyl cellulose is water insoluble excipient, but here in the water we can add some amount of ethanol plus some amount of acetic acid as specific ratio, so that ethyl cellulose also dissolves in the aqueous phase. Similarly, other excipients also can be taken. We can also take the help of acetic acid within the aqueous phase, so that the excipient dissolves in the aqueous phase. Similarly, we can take the commonly available excipient. There's a gum arabic or acacia. It can be taken in the deionized water at a specific ratio. And if it is required for the other excipients, even the required small amount of acetic acid can be employed to obtain the acidic pH, so that the few excipients dissolve well in the acidic pH. Similarly, if you take this PU or the polyethylene oxide also, it can be added to the aqueous phase deionized water, and then we can employ the electrospinning process. Now, if you take the other excipient like the PLGA, here it can be added to the organic solvent, that is a THF, that is a tetrahydrofuran, and we can add the NN-dimethylformamide. Similarly, if you are taking from the hydrophilic excipient from even the PVP K60 grade, here we can employ the anhydrous ethanol, and for the PLA or the polylactic acid, we can employ chloroform plus acetone at a specific ratio. So now if you are taking from even the chitosan as an excipient, here we can take the antitumor drug from the doxorubicin, and we can employ this technique so that we can entrap the antitumor drug within this excipient. Now the advantage of this electrospin sample is that it gives an extended release of this antitumor drug. Now, if you take from the chitosan has been employed for electrospinning, and the various types of the other excipients also can be employed. And whenever you are employing from the chitosan for the electrospinning process, here we can blend this excipient along with other excipients also, like the PU or from this polyethylene oxide, another from even this polyepsilon, from this caprolactone, the agarose. So the different excipients can be employed individually, or we can take about 2 excipients from the together along with the drug, and we can do the electrospinning process. So if you take the important applications of this from the chitosan is an important excipient, it has so many applications. In the electrospinning, we can obtain from the nano from the electrospin sample, and we can also obtain from this by employing this excipient. We can also prepare the nano from this drug formulations by entrapping the drug. We can also obtain from the encapsulated product from even the micro encapsulation of the micro spheres also can be obtained. And here by employing this excipient, various types of drugs can be employed and trapped. Either we can take the anti-asthmatic drug like theophylline, or the anti-infective drugs, or the anti-tumor drugs, or the antihypertensive drugs. So the various types of drugs can be entrapped by employing this excipient by the electrospinning process. And we can also obtain in the form from the nano formulations, or in the form from even the micro encapsulated product also can be obtained for the various types of the drugs. Now, if you take here from the electrospin nano from these formulations, they can be employed to entrap the various types of the drugs, and even the other organic compounds like the from this bioactive compounds, the growth factors also can be entrapped. Now, whatever the drug is entrapped within this excipient, the drug release has to occur. It will mainly occur by the erosion process and also by the diffusion process. So whatever the drug is entrapped within these excipients, normally the excipients will slowly undergo hydrolysis or the enzymatic decomposition, and the drug can be released slowly. In this way, you can get the extended release of the drug can be obtained. Example, the anti-infective drugs can be entrapped in the electrospin sample. So the various types of the anti-infective drugs can be loaded within the electrospin sample. Diller et al. Have prepared the electrospin nanofibers by employing the PLA or the polylactic acid and polyepsilon caprolactone. Either the single or the 2 excipients can be blended together along with the various types of the anti-infective drugs. So the various types of anti-infective drugs have been entrapped here. Example, amphotericin B is widely employed in the treatment of the infections, and these drugs, anti-infective drugs, has been entrapped, and it has been done for the in vitro evaluation, and the anti-infective activity is evaluated. Different type of the solvents have been employed so as to entrap the anti-infective drugs. Example, chloroform and even the THF, and the excipients are employed. We can employ from even the polylactic acid, polyepsilon, from even the caprolactone, either alone, or actually we can employ a specific ratio of the 2 excipients. So apart from the drugs, for example, we can also employ from even the silk from this, when this structure can also be employed. And if you have to electrospin it, the advantage that here is quite safe without any harmful effects, and the electrospinning of it can be done by employing a specific organic solvent, that is HFP, that is hexafluoro2 from the propanol can be employed. And the advantage with these structures is that they can be employed as scaffolds for the tissue engineering in the wound from the dressing. Whenever we are having some injuries or wounds, so the skin has to be treated, so it can be employed in the tissue engineering studies for the wound from dressing, or for the wound healing activity, it can be employed. So next is, if you take the how it all, they employed this organic compound from the calf skin. It was again dissolved in the organic solvent, that is a hexafluoro 2 from even the propanol, and then it was done for electrospinning. What are the electrospin from the scaffolds have been obtained? It is mainly employed for the wound dressing and for the tissue engineering studies. So for the wound healing activities or for injuries, these type of the electrospin sample can be easily employed on the skin. Similarly, now in the treatment of tumors, in the treatment of severe inflammation above the stomach, in the esophagus, sometimes if you have inflammation, and sometimes we can also have the occurrence of tumors. So here we can take a stent. Above the stent, we can apply the electrospin sample of the anti-tumor drug or the anti-inflammatory drug along the specific excipient, and this stent can be placed in the esophagus above the stomach, so that the anti-tumor drug will be released slowly and the tumors can be healed or the inflammation can be treated if I am able to entrap the anti-inflammatory drug. So here you can see the anti-tumor drug from when this paclitaxel was entrapped within this polyepsilon from the caprolactone above the bare stent. And the advantage of this process that here it will have the anti-tumor action. This anti-tumor drug, apart from the anti-tumor action, it's also having the anti-inflammatory activity also. So in the esophagus above the stomach, we can treat the tumors and inflammation by employing this technique. Next is, apart from the earlier drugs, we can also take the water soluble and from even this fat soluble compounds. From this vitamin B12, orally it can be absorbed, but because here the hydrophilic property, sometimes if you're employing high doses, the AUC value decreases. So as an alternative option, apart from the oral route of administration, it can be also employed the transdermal patch. So here the electrospin nanofibers of this vitamin B12 can be taken. It can be formulated as a transdermal patch. Here the toxicity is less, is a non-invasive approach. It can be easily applied on the skin so that the vitamin B12 loaded nanofibers, here the drug can be absorbed across stratum corneum, outermost skin barrier into the blood. So the advantage is that it gives a long-term application. Once the transdermal patch is applied, the drug can be slowly absorbed across skin to the blood, so it can give a long-term action. So we can take both we can take the vitamin A and vitamin E. It can be also entrapped in the transdermal patch by employing the electrospinning sample by the celluloestate excipient. So that we can take celluloestate and by employing electrospinning, the vitamin A and vitamin E can be entrapped, and then this can be fabricated as a transdermal patch. Then if we do the in vitro drug release studies, it is observed that within about 6 hours, it shows a slow drug release, and at the end of the 24 hours, it gives a slow extended release. So compared to the films, here the transdermal patch can give an extended release. It can be applied on the skin, so the drug can diffuse slowly across stratum corneum into the blood, and it gives a long-term action of the drug. So this electrospin nano from this fibers, we can entrap various anti-inflammatory drugs also, like from the naproxen by employing various excipients like the chitosan, polyvinyl alcohol, polyacrylic acid, polyepsilon from even the caprolactone. The advantage with this is that it gives a rapid onset of action. It can be easily employed and can avoid the extensive hepatic effect. Here, in place of oral route of administration, we can employ from even the sublingual route of administration for this nano from the structures. When you are placing it under the tongue, here initially the drug dissolves fast. It gives a prompt onset of action. Afterwards, under the influence of the excipient like polyvinyl alcohol, the drug release is slow. It can also give an extended action up to 1 day. Razia Begum et al. They have employed the electrospinning technique so as to enhance the dissolution rate of the poorly soluble drugs like anti-inflammatory drugs by employing a hydrophilic excipient in the ratio of 1:4. The enhancement of the dissolution rate of this anti-inflammatory drug was achieved by electrospinning technique. It was accepted in the advance in the natural sciences from the journal. Zia Shen et al. They employed from the anti-inflammatory drug from this diclofenac sodium for electrospinning. These mainly employed so as to achieve the drug release in the area from the colon. So in the area from this colon, if you have the inflammation, by entrapping this anti-inflammatory drug, it can be treated. In the in vitro release studies, it was seen that in the acidic pH, the drug release was very less, but in the alkaline pH of the rectum, from the drug shows extended release. So it can be employed in oral formulation. The drug release occurs mainly so as to treat the inflammation in the area from the colon. We can treat the inflammation by employing this technique. Simvastatin electrospin nanofibers were prepared for the oral extended release. The development of from this Simvastatin and electrospun nanofibers is a novel approach for the extended release, and it was here we obtained this article in the journal of from this pharmaceutical investigation. So the various types of drugs and excipients which are employed by electrospinning so as to obtain the various immediate release type formulations. So by employing this electrospinning for the water insoluble drugs, the drug dissolves fast. It can give a prompt onset of action. It gives a quick onset of action. So various types of the drugs like anti-inflammatory drugs like the oxycams and even the statins, which are employed so as to decrease the lipids in the blood. These are water insoluble drugs. They can be entrapped within this from the polyethylene oxide or from even this other type of hydrophilic excipients can be employed like the PVP. So for these water insoluble anti-inflammatory drugs, the drug release can be enhanced as an immediate release formulation by employing the electrospinning technique. Similarly, aceclofenac also can be entrapped within the PLA, and here we can do the electrospinning for the immediate release of the drug. Similarly, other drugs also like the acyclovir, antihypertensive drugs and anti-infective drugs like from this ciprofloxacin, and again other anti-inflammatory drugs like from this moxifloxacin, ketoprofen, they have been entrapped in the various types of hydrophilic excipients and by electrospinning technique so that we can get the immediate release. And the advantage of this electrospinning technique for the water insoluble drugs is that it will enhance the rate of the drug release, so it can give a faster onset of the action. So apart from the immediate release formulation, electrospinning technique can also be employed for the extended release formulations. Various types of excipients can be taken so as to entrap the drugs which are having low elimination half-life, and by employing this technique, we can extend the drug release. So the various types of drugs like anti-infective drugs or the anti-tumor drugs or even anti-infective drugs like the amphotericin B, they have been entrapped by employing various types of excipient like from the PLGA, PVA, cellulose acetate, polyepsilon from this caprolactone. So the advantage of this technique is that it can give an extended release for a higher, it can give a long-term action. So the various types of drugs have been entrapped for the extended release. Example, anti-tumor drug from this doxorubicin was entrapped by employing the excipients like the PLGA, and when the PVP we can employ it when the anti-inflammatory drug also. So here we can see so many different classes of drugs have been employed by electrospinning. Example, anti-tumor drugs, and by employing various types of excipients. And even the anti-infective drugs have been tried by the various types of excipient like the rifampin, even from the drugs like the doxycycline. So various anti-infective drugs and anti-tumor drugs have been tried for the electrospinning technique by employing various water-soluble and water-insoluble excipients, either we can employ alone or we can add the two excipients together, both the water-soluble and water-insoluble excipients, so that we can optimize the drug release up to the required number of hours. So here again, we are employed from this PLGA and then from the PG plus PLA, so as to entrap the various types of antiviral and other anti-infective drugs. Similarly, antihypertensive drugs have been entrapped in the PLGA, and some anti-inflammatory drugs have been entrapped by taking the various types of the excipients. So apart from the drug release studies from the electrospinning is also an important application in the area of the tissue engineering studies. The tissue engineering deals with from the development of the various types of electrospin sample so that we can replace the disease or the damaged tissue in the humans. Example of tissues which can be treated by tissue engineering include the skin, heart, or even the bone. So for example, if you're having the defective bone, defective skin, etc., it can be treated in the case of the tissue engineering studies by employing a specific electrospin sample. So in the case of this bone tissue engineering, here we can obtain the electrospin nanostructures. These can be employed to heal the various types of tissues by employing the bone tissue engineering. Now here you can take the various types of the electrospin formulations are available. Example is the AO's trade name. It can be employed for the wound healing activity. It contains from this non-woven structures, and this mainly employed for wound healing application. Similarly, the rethink from the surgical mask. Here it can be employed as a surgical mask so that we can prevent the inhalation of the impure air. Similarly, the nano dream from the textile industry, from the nano layer has been obtained so that above the cloth also can obtain, we can employ this electrospin sample. Similarly, the Bria safe in the air filtration, these structures can be employed as a mask so that it will prevent the inhalation of from this microorganisms or the toxic gas or the dust. Similarly, in the tissue engineering studies, also these nanofibers have been employed by scaffolding for the growth of the tissues, mainly employed for the wound healing activity, wound dressings, because here whatever the electrospun sample we are employing, it will have the space for the activation of the tissue. It will increase the oxygen exchange in addition to the reinforcement. In the air filtration also it can be employed. The electrospun sample, apart from cross-linking, it has a spatial structure, provides the ability to capture the small size particles and filter the environment. So in the air filtration also it can be employed apart from wound healing activity. In the defense industry also, we can prevent the inhalation of the toxic gases. These electrospun samples, it can entrap the toxic from the stimulants. So we can prevent the inhalation of the toxic gases. So the electrospun sample can also be employed as a face mask, Swasa face mask, odorless plus, and it can be employed as a device recommended for the doctors. Similarly, Swasa surgical mask, it can be employed as a medical device as a surgical face mask. And when this ultraspun sample has been obtained from the biotronic industry, here the nanofibers, it can be applied on the stent surface as thin, highly elastic from the membrane can be obtained, can be employed in the stent. Similarly, the Urgi clot, here the dextran sample can be employed for electrospinning. This is employed in the tissue engineering studies, that is in the case of bone bleeding, so as to overcome the defective bones. Surgical implants and wound dressings, bone void, defect filling material. The various components are employed like the beta tricalcium phosphate, bioabsorbable polymer, and SIV containing from the CaCO3 can be employed. This will be employed for tissue engineering studies so as to improve the growth of the bone. Similarly, the Heal Smart, it can be employed as an anti-infective dressing with the addition of the HA, the hyaluronic acid, so that it can have the anti-infective action. So the commercial electrospun products for the healthcare, it includes the beta TCP, beta tricalcium phosphate, SIV, that is a siloxane from this containing CaCO3, BioWeb. For the clinical use, it can be employed as a PTFE vascular stent coating. Similarly, we can also employ it can be obtained from the industry, that is a Biotronic, FDA approved in 2018. Here, it can be employed as a stent. Similarly, the Ribosys, FDA has cleared it in the USA in the year from the 2018. It can be employed as a bone void filler in the case of the tissue engineering studies. Similarly, if you take this Surgiclot, for example, the clinical use of the dextran mainly for the wound dressing. So we can see in this slide the various extracellular matrix proteins that are found in the bone, like the type 1, type 5, osteo from this calcin, osteonectin, fibronectin. When the caprolactone, silk fibrin, and when from the ketose has been employed, and the inorganic excipient like the hydroxyapatite, fluorohydroxyapatite has been employed either alone or you can combine two excipients. By employing these excipients, it is employed for electrospinning for the tissue engineering studies. Here we can obtain the electrospin nano from the structures, thread-like structures, having a diameter of 50 nm to about 1000 nm. And these can be employed in the tissue engineering studies so as to overcome the problem of the defective bones. Here the bone will be formed at the end of about, say, it takes about, say, 3 to 4 weeks so as to form the required bone. So in the case of the bone tissue engineering studies, these electrospins of that. And similarly, you can take from the gelatin plus polyepsilon caprolactone, polylactic acid, bone from even this bioactives like the inorganics, hydroxyapatite, fluorohydroxyapatite, silica, and even the CaCO3 and the calcium phosphate. This will improve the bone extracellular matrix ECM. Similarly, we can also take from even this polysilane along with the CaCl2. So these type of the electrospun samples, by employing the different, we can take a blend of the different excipients These have been employed in the case of the tissue engineering studies, mainly to improve the growth of from the tissues involving mainly the defective bones. So here electrospinning is a very important technique. Here you can obtain the highly from this nanofiber structures and the required pores, and by employing the various types of the natural polymers, inorganic or the organic polymers can be employed. And in this technique, we have to adjust the various points like the humidity, absolute from this, even the temperature, how much voltage you're employing, so that we can obtain the required nano sample can be obtained. So by altering the various points like the how much amount of excipient we are employing, how much amount drug we are employing, the drug excipient ratio, the molecular weight of the excipient, the viscosity of the value from when we can employ the required voltage. We have to adjust the rate of flow of the solvent blend, and we have to also adjust from the distance from the glass from the syringe up to the drum-like structure. The electrospin from the nanostructures can be obtained, and it can be employed in different applications like the air filtration, engineering studies, from the textile studies, and even the electrical engineering also it can be employed. So these electrospin nanofiber scaffolds are thin from this membrane-like structures. They are very important applications in almost every field, including in the case of the clothing, or in the case from if you take even the cosmetic formulations or the tissue engineering scaffolds, various types of drug formulations, immediate release or extended release formulations of various types of drugs. Either it can be anti-tumor drugs, anti-inflammatory drugs, anti-infective or anti-hypertensive drugs, and when for the wound healing applications, it can be done. So electrospinning is here, it plays a very important from this role in the different fields. The electrospinning technique has exhibited excellent role in the various fields, mainly in the case of the tissue engineering. So all these are from, you can see the references. Thank you. So if you have any doubts, you can ask. Hello, sir, you are able to hear me? If you have any doubts, you can ask. Thank you, sir. So actually we can stop here, I think. In that case, I will stop here, sir. Thank you, sir, for organizing this FDP.
Sir, in group chat, they have asked some questions, sir.
Okay, so you can just announce it, sir. In that case, I'll answer.
Apart from silk, fibron, and any other material can be used.
Sir, mainly it can be employed in case from the tissue healing. So we can take any organic from this. Suppose if it is having any important role in from this tissue healing, it can be employed, sir. You can see in the internet from the various types of excipients which can improve from the tissue growth. It can be taken up for the electrospinning.
Okay, sir. One more question is there, sir. Challenges faced in nanofiber formulation.
Yes, sir. Here the main point is that whenever we install this electrospinning equipment, it should be installed only in the AC room. Suppose if you're not employing the AC room, if you're employing the ordinary room only, then the main point is that here we have to adjust the humidity and from even the temperature has to be adjusted, sir. Because in the month of October or in the month of October or November, the humidity, if you see, is very less, even the temperature will be less. Whereas in the month of April or May, the temperature and humidity increases. So if you alter the value of temperature and humidity, obviously it will affect the efficiency of the electrospinning process. So if you want to have throughout the year, if you want to have the same type of electrospin sample, you require to install the equipment in the AC atmosphere.
We had no book when we are studying. We were saying the books were not there.
Okay, sir. Sir, one more doubt is there. What is the cheapest equipment available in the market?
I would say it will take at least around 8 lakhs, sir. Actually, at least it will be 8 lakhs, sir. The address will be available. It will be above 8 lakhs only.
Okay, sir. Okay. I think there are no further doubts, sir.
Okay, sir.
So on behalf of our management and organizing committee and faculty, I thank our Professor and Dean, Dr. M. Mohan Varma garu, for sharing these valuable insights about this topic, and thank you so much, sir, for sparing your valuable time.
Thank you, sir. Thank you, sir. I am leaving, sir. Thank you very much, sir.
Thank you. Thank you.
So now I request
Yeah, good morning. Good morning, delegates. On behalf of management, principal, and organizing team, it's my privilege to propose a vote of thanks on this occasion. My heart fills with lots of gratitude and respect for our distinguished guest speakers, Mr. R. Vijay Bhaskar Reddy, sir, Director and CSO of Theralen Pharma Private Limited, Hyderabad, and Dr. B. Chandrasekhar Reddy garu, Professor and HOD of Vagdevi Pharmacy College, Warangal, Dr. P. Bhargav Bhushan Rao, sir, Professor and Principal of A.M. Reddy Memorial College of Pharmacy, Guntur, Dr. G. Ramana Reddy, sir, Professor and HOD of KVSR Siddhartha Pharmacy College, Vijayawada, Dr. M. Mohan Varma, sir, Professor and Dean of Sri Vishnu College of Pharmacy, Bhimavaram, for accepting our invitation and sparing their valuable time for enlightening us with their commendable talk on exploring new innovations in novel drug delivery systems. Without your kind presence in the FDP, we could not have accomplished what we had dreamt for. Thank you all for clearing our concepts and enhancing our understandings. I'd like to thank our respected management, Secretary Sir CHBV Subbarao Garu, and Technical Director CH Apparao Garu, and our torch bearers Principal Dr. V. Bhaskar Raju Sir, and Organizing Secretary Dr. P. Narayan Raju Sir, and other organizing teams for leading and inspiring us in working towards the goal of conducting this FDP. Thank you, delegates, for registering and making this FDP a huge success. Thank you all once again. This is Shravanti, Assistant Professor from Srivasavi Institute of Pharmaceutical Sciences. Thank you.
So thank you, sir. We will sign off.
Thank you, sir. Thank you.
Fine.